Well, hello everybody. Welcome to your sex linkage video, where we'll review the sex linkage lecture and just go over the few topics that the majority of you reported to be confusing. And for this lecture, that really comes down to dosage compensation and X-inactivation. We'll spend a good amount of this video reviewing those two topics at the end. The other thing, though, that came up in a few of the reflections was an interest in reviewing the information relevant to just the transmission of sex-linked traits, just diagramming a few sample problems, kind of like what we did last week, where we just kind of went through some crosses to see how they would work. So let's do that first. That way we understand how to track sex-linked traits and how to represent them in Punnett squares and predict offspring from them and all of that good stuff. So first, let's consider the most common example of a sex-linked trait, and that is of course a mating between a man and a woman, so XY and XX, no surprise there. But let's go with color blindness and say that it's a colorblind man mating with a woman who has no color blindness in her family at all. So she's homozygous dominant for normal vision. And I'm using B here. One of you actually even recommended this, going back to my idea that C's are hard to tell apart, especially when they're superscripts on the 6 chromosomes here, so I'm just using B's to represent color blindness. So any X chromosome that has a dominant B has normal vision, and any X chromosome that has a recessive B is colorblind. So to diagram this cross between a colorblind man and a full-seeing woman, a woman who's not colorblind at all and has no history of colorblindness in her family, would be diagrammed as a simple Punnett square. So we'll set up the Punnett square. We'll put one parent on the top of the square and the other along the side. It really doesn't matter which parent you put where. There's no right way to do this. And then we're going to treat this like a normal Punnett square, where we're just going to bring down and bring across all of the alleles that we see. And we would have that outcome. So what we see is that 100% of the children of this mating have normal vision. They can all see color. But 50% of the children are carriers. Another way to say that is all of the daughters are carriers. Carriers are individuals that have a recessive trait but don't show the recessive trait because there's a dominant allele there as well. Little side note on that is we would never refer to a male as a carrier, because for sex-linked traits, you either don't have the trait because you don't have the allele, or you do have the trait because you do have the allele. A carrier means you have the recessive trait, but you're showing the dominant phenotype. So only females can be carriers. All right, so let's take one of these daughters and fast forward her life a bit. So she's been seeing color all her life, but she's a carrier. She knows she's a carrier because her father's colorblind. And she meets and falls in love with some guy who's got normal vision, so we know he must have the normal dominant allele. And they get married and they have kids. And let's diagram their cross. So there she is, our color-seeing carrier woman, and her man, the normal-seeing man that she's having kids with. And they can have daughters. Half of their daughters will be normal-seeing and have nothing but dominant alleles. And half of their daughters will be carriers, but all of their daughters will be able to see color. Excuse me while I clear my throat. And then of their sons, half of their sons will see color. But there it is. Half of their sons will be colorblind. They're getting that color blindness from their mother. They're getting that colorblind trait from their carrier mother, because for sex-linked traits, the fathers contribute nothing on the X chromosome. Though dad can see color and dad has a dominant allele, dad is not able to pass that dominant allele on to any of his sons. Well, you say, well, what about the sons that got the dad's X chromosome? Well, they ain't sons. They're daughters. Because as soon as dad gives his X chromosome, he has allowed a daughter to be formed, not a son. The only way sons are born is if dad gives his Y, and if dad gives his Y, the only information those sons get on their X chromosome come from their mother. I'll use myself as an example. I can see color solely because my mom gave me her X chromosome that had the normal dominant color blindness gene or allele on it. Though I am a man because of my father, I'm a male because my father gave me his Y chromosome. All of my X-linked traits come exclusively from my mother. My father didn't give me his X, because if he did, I'd be his daughter, not his son. So that's kind of how we think about colorblindness. Now, every once in a while, we will have a colorblind, we will have the daughter of a colorblind individual in our class. So that might pertain to some of you. If your dad is colorblind, then we know your father must be this. And if you are your father's daughter, he gave you his only X chromosome, you are unequivocally a carrier. And if you have children with a man who can see color, he will be unequivocally dominant. And if that's the case, all of your daughters will be able to see color. But statistically, half of your sons will be colorblind. And that's okay. Being colorblind is not all that limiting. It's nothing to be upset about. But we know it to be a genetic fact that if your father is colorblind, each of your sons has a 50% chance of being colorblind if and when you have children. So just an introduction into sex-linked traits using colorblindness as an example. We'll come back to colorblindness near the end of this video, but there's one more cross that I'd like to show you just to drive this home. So there are colorblind women out there. They're far more rare because in order to be colorblind as a woman, you need to have 2 recessive alleles. In other words, your father needs to be colorblind and your grandfather on your mother's side also needed to be colorblind. Colorblind women have colorblind fathers. Oh, you can't really see that fully, so let me just adjust here. Colorblind women have colorblind fathers. That's where the first X chromosome came from. And they have colorblind or more likely carrier mothers. And that's where the second X chromosome came from. So that's possible. It's just rare. If a colorblind woman marries a normal seeing man, all of the daughters they have together will see color. They'll be carriers, but they'll see color. Interestingly, when a colorblind woman has sons, all of those sons will be colorblind. Because dad is giving his Y chromosome, the only source of the X chromosome for these sons is the mother, and the only X chromosome the mother has to give is a colorblind X chromosome, so all of those sons will be colorblind. So hopefully that's a little bit more clarity on diagramming these sex-linked trait crosses, as well as just understanding the transmission genetics around them, how these traits are transmitted genetically. So let's move on. Excuse me again. Let's move on to X-inactivation and dosage compensation. So we're right back to where we started. Males and females. Males have 1 X chromosome. That's what determines gender in males. Females have 2 X chromosomes. That is a dosage problem. We think of genes and alleles as being the instructions for building proteins. You've heard that already in general biology, and we'll come back to that after Mendelian genetics when we hit molecular genetics hard. Genes are the instructions for building proteins. Alleles are the instructions for building proteins. So, how much protein a cell has inside itself is in some way proportional to the amount of alleles there are for that protein. In other words, if you have 1 working allele and 1 mutant allele, you'll have half as much protein in a cell than a cell that has 2 working alleles. That's where we get at dosage. Now, for the most part, our protein levels are balanced across the species because the differences in these levels are pretty minute. But we've got a big problem with the X chromosome as a species. Left the way I've diagrammed it here, for every single possible gene on the X chromosome, and there are many, there would be 2 times as much protein for all of those genes as compared with males. Because males have 1 of each of those alleles, and females have 2 of each of those alleles. And doing that would create a massively out-of-whack balance where females have far too many proteins than males do. In fact, not to be overdramatic about it, but it's fair to say that might not even be the same species, because we would essentially have a different chromosome number. Not good. We can't have that. So what we need to do is we need to compensate for this dosage imbalance. We need to have dosage compensation. We need to find a way where we can get the amount of protein in these two members of the species equal. There's really only two strategies you can do. You can add an X chromosome to every single male, so now we have an equal amount of alleles for the X chromosome genes and an equal amount of protein. But then the question becomes, well, how do you do that? Each parent contributes one chromosome for each pair. How are you going to get a third chromosome in for only the sex chromosomes? It's going to require non-disjunction and meiosis, right? And that's actually something that causes the cell cycle to fail. So that's not going to work. We can't do that. Males, as may be sometimes usual, kind of muddying the waters here because we're so locked into what we're doing. Alright, so the only other choice we have to compensate for this dosage imbalance is to somehow figure out a way to get rid of an X chromosome in women. And if we can do that, we've balanced the scale the other way. We've given women only one X chromosome to work off of. Males already have only one X chromosome to work off of. And now we have compensated for that dosage imbalance. So that is an easier thing to do. Not easy, but easier to do. And that's what we do in human cells. We get rid of an X chromosome in every single female cell. So the question becomes, well, how do you do that? How do you get rid of an X chromosome? And the process is called X-inactivation. So now we see how these two terms are linked. Many of you, in your final question for the lecture video, said you weren't too sure about dosage compensation, and you also weren't too clear on X-inactivation. We've just linked it. Dosage compensation is a strategy, is a way of thinking for dealing with the dosage problem between the sex chromosomes in the different sexes, males and females. We need to compensate for twice as many dosages of X chromosome genes compared to one of each dose in males. That's the dosage compensation. How do we achieve dosage compensation? What is the mechanism for achieving the strategy of dosage compensation? X-inactivation. So X-inactivation achieves dosage compensation. X-inactivation is the mechanism to realize the strategy of dosage compensation. We need to compensate for this different number of X chromosomes between the sexes, and we use X-inactivation to do it. So what X-inactivation involves, I try not to get too much in detail here, but it's a really strange story. There's a gene on every X chromosome called X-ist, X-I-S-T. And this gene tries to express itself. What we mean by that will become more clear when we go over molecular genetics. But suffice it to say right now, we just start making an RNA molecule of this gene. And all X-ist genes try to do it. But these RNA molecules have a very peculiar and interesting property. They can sense the RNA molecules around them with the same sequence. So since this one is a little bit longer, this stops expressing. Since there's no messenger RNA at all, since there's no RNA at all here, this stops expressing. This X-ist molecule is winning the race, and it keeps expressing. It actually expresses itself to be a very, very long molecule, far, far longer than any normal or typical RNA molecule. In fact, they're even given a special name called long non-coding RNAs, which becomes important as you move further up in your curriculum to courses like cell and molecular biology. But the end result of that is that this RNA that encapsulates and covers this X chromosome begins to wrap this X chromosome up very tightly. The DNA in this X chromosome begins to compact, like chromatin, but very, very dense chromatin. And that X chromosome that won the X-ist race compacts itself so densely and so compactly and so tightly that the DNA in this tightly wrapped X chromosome becomes inaccessible. The genes can't even be recognized. They can't be expressed. Proteins can't be made from this X chromosome. And so that means that this X chromosome has become inactivated. Now, the only X chromosome that the cell has to use, the only genes that it can access and express to make the proteins from the X chromosome, are on this X chromosome that was not inactivated. This thing here is so dense that you can actually see it on a light microscope. And Murray Barr, a not entirely successful early biologist, noted these. He realized that they were only present in female cells. He wasn't quite sure what they were, but being perhaps a typical male scientist, he named them after himself anyway. So these are Barr bodies. What is a Barr body? A Barr body is an inactivated X chromosome. A Barr body is an X chromosome that is wrapped so tightly that the genes on it can't be used by the cell. A Barr body is an X chromosome where Xist, this long non-coding RNA, was able to wrap it very, very tightly and silence it. A Barr body is the cell's way of achieving dosage compensation. To be fair to the history of the Barr body, not too long after Murray Barr noted these and published it and named it after himself, Mary Lyon, a pioneering female scientist in the life sciences, began studying Barr bodies, and she figured out what they actually were. It is Mary Lyon that we have to thank for the notion of dosage compensation and the mechanism of X-inactivation. Not the molecular mechanism, but at least having an understanding of what it was and why it was happening. Mary Lyon was an incredible female scientist. She made many, many contributions to the life sciences. And she also felt it was very, very important for women to have a doorway into STEM fields, which was a pioneering thought that many years ago. And so, believe it or not, not only did Mary Lyon explain what Barr bodies are, but later on in her career, she went on to found one of the most prestigious female colleges still in existence today. She was the founder and first president of Mount Holyoke College, not too far from us here in Western Massachusetts. Okay, so now we have a little bit more of an understanding of what X-inactivation is. X-inactivation is a mechanism of dosage compensation where an X chromosome is wrapped so tightly and so densely that it is inaccessible and unusable by the cell. So female cells only have one X chromosome to run on, and they're only making one concentration of each of the proteins from that X chromosome because of the presence of this Barr body and because of X-inactivation. Males, of course, only have one X to begin with, and they're only making one concentration of those proteins anyway, because we only have one X chromosome, and that strategy compensates the dosage between male and female cells, making us, once again, one species capable of one single physiology and one homeostasis. Everything is in balance. But there are implications here. I'll quickly review the calico cat example, and then we'll go on to talk about a different example that's actually, in my mind at least, a little bit more interesting. So, for calico cats, let's imagine that cat in kind of an embryonic stage, so just a blob of cells really, but there's many, many cells in here, so I'm representing an early embryonic stage, but definitely multicellular. And let's imagine that the cells that we're looking at here are going to be skin cells. So I'll just draw a couple. And let's also imagine that this cat is a female cat, and she is heterozygous for the ability to express fur color. Fur color. So if they're dominant, those cells express color, meaning you get a patch of colored fur. However, the recessive trait is the inability to express fur color, or essentially, just to kind of simplify it, an albino patch, a white patch of fur. Again, a female cell, a female cat, this is an X-linked trait, and because this is a female cat, we are going to experience X-inactivation. The difference is that X-inactivation is random, and it happens fairly late in the developmental process. Not in the lifetime, but in the developmental process, so fairly late in the embryonic development. So essentially, randomly, half of the cells in this female cat's skin will inactivate the recessive chromosome, the chromosome carrying the recessive trait. But equally randomly and equally likely, another half of cells are going to inactivate the chromosome that has the dominant allele. So all of these cells will express fur color, and all of these cells will express white fur. Just trying to reposition my camera. And it happens randomly, relatively early on in development. So what I'm going to do, just for the sake of simplicity, is every single cell that is using the dominant X chromosome and can express fur, I'm going to represent as a filled-in cell. And I'm going to leave the cells expressing the white patch chromosome as white. So it's random. So this cell might express the recessive carrying chromosome, and this one does too. Maybe this one does, and this one does. And we get this kind of patchwork hybrid pattern of expression. Now, at this point, we've kind of locked in which X chromosome is going to remain inactive and which isn't. So as each of these cells continues to divide further on in embryogenesis, the cells that arise from them excuse me, are going to have the same X chromosome inactivated. So now, as I use my artistic skills, which I know you are all completely envious of, and I draw myself a cat, this cell here gave rise to a patch of skin in the cat that expresses fur color. This one cell, as it continued to divide and divide, and all of the daughter cells from it had the recessive carrying X chromosome inactivated, all of those cells are expressing the dominant trait, which is color. Just next to it and above it, this cell created a patch of skin cells that can express color, and that's white skin cells. This skin cell made another patch of color. This skin cell made another patch of white. We have white below. We have color next to that. And as you can see, I'm drawing a calico cat. So most of the time, a true calico cat, a cat that has patches of tabby fur and patches of white fur, almost every single time you find a true calico cat with that kind of patchwork pattern, you're going to be looking at a female cat. So you can gender identify many, many cats just by looking at their fur pattern because of X-inactivation. Are there calico males? Technically, no. Genetically, there are no calico males because it requires X-inactivation. But there are fur coloration patterns in cats that look calico, that look patchwork, but they're actually due to differential division of cells during embryonic development, and that's a little bit of a different story. So there are times that you might misidentify a cat as calico, and it's a male cat, but that's not a calico cat. The patchwork pattern is subtly different if you look closely. So that's the example I give in the lecture, but let's take that to a different example that's more relevant to us. Let's go back to our colorblind carrier female. Now this female has normal color vision, right? Because she has a dominant allele, she has normal color vision. In other words, when she was in elementary school and she had to go to the nurse's office for a lice check, and while she was there, the nurse put strange glasses on her and gave her a little book. And in that book, there were colored dots. And some of the dots were red, and the red dots made a 5, and some of the dots were blue, and the blue dots were the background. And the nurse said, "What number do you see?" And that little girl said, "I see a 5. And the nurse said, "Yes, very good, go back to class." That was a colorblind test, and she passed it. This girl sees color. That said, if we take a look at the eyeball, and now what I want us to do is imagine that we're looking in the back of the eye. So here's the pupil of the eye, right? And the iris and everything that we see. At the back of the eye is the retina. That's where we actually collect our vision information. That's where the light projects in an eyeball, and that's where we process vision stimulus, vision inputs. So I'm going to take this patch of cells, and I'm going to represent it here. So here's that patch of cells, right? I'm just using my hand to kind of represent it, and now we're looking back into the retina. So the pupil would be here, and we're looking at the back of the eye at the retina. The retina is made up of a lot of retinal cells. These are specialized neurons. They detect color, and light. So just like that embryonic cat, here is that woman's eye, her retina. And she's a woman, so she's going to experience X-inactivation. Sometimes when she makes a bar body, what will be spared is the X chromosome carrying the dominant allele. But sometimes when she makes a bar body, it's going to be the X chromosome coloring the colorblindness allele that is left as accessible. I'm going to represent these cells as filled in and these cells as blank. X-inactivation is random. And so this represents her retina. This represents the back of this woman's eye. Trying to go for about 50-50. It looks relatively even to me. So really what we mean here is in this woman's retina, this cell can detect color, this cell cannot. This cell is colorblind, this cell can detect color. This cell is colorblind, this cell can detect color, this can detect color, this one's colorblind. Just like the calico cat's fur is a patchwork of albino and colored fur, this woman's retina is a patchwork of color-seeing and colorblind retinal cells. Well, we know she can detect color because she passes the little test that the nurse gave her. She can see the red dotted 5 in the background of blue dots. She's not colorblind. But we can make the prediction, based on our understanding of X-inactivation, that she doesn't see colors as vibrantly as you and I. In other words, the intensity of her reds and greens are probably different than the intensities to you. So I did this experiment once, believe it or not. I had this amazing insight, and I realized that this was the case for women, and I realized I could do the experiment to prove the prediction. And so I knew a woman, a girl, who had a colorblind father. So I knew she was a heterozygote, because if her father was colorblind, he gave her his only X chromosome, which we know was a colorblind chromosome, but she could see color. So she wasn't colorblind. So I knew she was a heterozygote. And I went up to her and I said, I'm going to do an experiment. I have a very important question for you. And she said, okay, fine. And I said to her, do you see reds and greens as vibrantly as I do? And do you know what she said? She said, how the hell should I know what reds and greens look like to you? So, of course, I didn't do that experiment because there's no way to. This gets at the notion of perception. We actually don't have any idea what the real world is like to any other one of us. I know what red and green and blue looks like to me. I know what fresh baked bread smells like to me. I know the smell of a sunny spring day for me. I don't know if you experience those stimuli the same way. For all I know, what you actually sense with fresh baked bread is completely different than what I sense. But all our lives we've walked into a room, and when we've smelled bread being baked, we said, someone's making bread, even though our perceptions of that may be different. I don't know what red looks like to you. I don't know what green looks like to you. But we can both point to a leaf and agree that whatever we're experiencing from that leaf, we call green. It doesn't mean we're seeing the same things in our brain. So these heterozygous carrier females for colorblindness see color in the sense that they can distinguish red from green. They can distinguish those two colors apart from one another. They can pass the colorblind test. But whether their reds look the same as they do to us who have one dominant allele, or for females too, whether the intensity of the colors or the shades of the colors or the hues of the colors are different for those carrier females, well, I'm quite sure genetically they are different. I just can't tell you in what way they're different because I don't know what it's like to experience the world through their eyes. So, not to get a little metaphysical on you there, but I do think it's important once in a while to note that we really don't know what reality is like to any other one of us. We only experience the world through the limitations of our own 5 senses, and we assume because we have consensus that we're all experiencing the world the same way. But just because we all agree on what green is doesn't mean we all see green the same way. So, hopefully that serves to clarify those two concepts for you. I hope that those crosses that we did with the Punnett squares not only clarify how sex-linked traits are inherited, but also just give you a sense of how to diagram them using a Punnett square and how to predict the offspring of sex-linked traits using a Punnett square. And I also hope that we helped to clarify dosage compensation as a concept, X-inactivation as a mechanism, the reason why it has to happen, what a bar body actually is, and finally, the implications of X-inactivation on the expression of phenotypic traits in females rather than males. And let me catch myself, because there was one other thing that a few of you mentioned, but it's probably worth pointing out here, which was triplo X, females that have 3 X chromosomes, so essentially Down syndrome, but not on the 21st chromosome, on the X chromosome. We know that Down syndrome has many phenotypes associated with it. We know that some of the others, like Patau syndrome, have many phenotypes associated with it, but women with triplo X have no ill effects whatsoever, and it's been a big question as to why. The answer as to why is X-inactivation. X-inactivation. We have this mechanism for the Xist gene to be turning off other X chromosomes. So in triplo X females, we want one X to work. That's the dosage compensation strategy. Men have one X, so women's cells get one X to work, and all of the other X chromosomes, typically 1, but in this case 2, all of the other X chromosomes get tightly compacted and be made inactive, made inaccessible, in order to achieve the dosage compensation. So now there's 1 X chromosome in this female cell and 1 X chromosome in the male cell, and we have an equal dosage. So the reason why triplo X females show no phenotype is because their cells are actually running on 1 X chromosome, just like any healthy female cell is. We can make one distinct prediction, though, can't we, for triplo X females? And it's there on the screen right in front of you. A triplo X female should always have 2 bar bodies in her cells because X-inactivation took both of the extra X chromosomes and wrapped them up tightly. All right, that's it. That's plenty. I won't keep you any longer. Please let me know in the comment for this video if this was clear for you. And of course, reach out to me with an office hours appointment or for a little bit of extra one-on-one over Zoom if you'd like any additional help with this material.